very clearly defined, and you can see almost the nuclear membrane that surrounds it. You do not see chromosomes inside the nucleus. The nucleus is dark because the chromosomes are still in long, thin strands called chromatin threads, or chromatin. On this particular cell, we can't see the centrioles, but under your microscopes, you may be able to see these two little dots outside the nucleus. This cell is in interphase. Remember, interphase is not a true phase of mitosis. And since most of these cells on this particular view are not undergoing mitosis, the majority of these cells are in interphase. Following interphase, the nuclear membrane begins to break down, and the individual chromosomes begin to condense or shorten. This illustrates the first true phase of mitosis, which is prophase. In this thickened and shortened condition, the chromosomes can easily be seen through the microscope. The nucleoli, the spherical staining bodies within the nucleus, which were initially present at interphase, have disappeared as the chromosomes thicken. At the end of prophase, the chromosomes appear to be randomly distributed. They're irregularly arranged in the cytoplasm. The centrioles, which were initially located just outside the nucleus, separate and move to opposite sides of the cell. In this diagram, we can see illustrated by the dotted line the fact that the nuclear membrane begins to break down or disappear. The nucleolus that was present during interphase also begins to break down and disappear. The centrioles, which were together here, one remains, the other has separated and moved down towards this side of the cell, and in the process forming these fibers, spindle fibers here and aster fibers here. The chromosomes that were long, thin strands have now become visible and are randomly distributed in the nuclear area. Later, during prophase, the centrioles, one located here, the other here, are on completely opposite sides of the cell. The spindle fibers extend all the way from one centriole to the other. They pass through the chromosomes. If we take a look over here at a blown up view of what is occurring here, here's a chromosome. This whole structure here and here together make up a chromosome. Each half of a double chromosome is called a chromatid. These two chromatids are held together in the center by a structure called a centromere. During prophase, a spindle fiber is attached at the centromere of each chromosome. So, in review, a chromosome is actually two chromatids, in other words, two identical strands of DNA held together by a centromere, this little button. And during prophase, the spindle fiber attaches to that centromere of each chromosome. On some real cells, this cell is probably in prophase. The nuclear membrane is not as intact. This cell, you notice the edge of the membrane along here is beginning to break apart. Here you see that the dark area in the center is not smooth and homogeneous like it was in the interphase cells. This cell is in prophase. Here we have a good cell in prophase. You can see the breaking up of material in the nuclear area here. Here we have a cell in prophase where there is no smooth nuclear membrane around the nucleus, and the chromosomes, the very dark little specks in here, are becoming very visible. The next stage is metaphase. In this stage, the chromosomes are located across the middle or equator of the cell. Their positions are highly organized. A spindle of fibers radiates from opposite ends of the cells. However, it is not observable with your microscope. Now, in metaphase, the spindle fibers, which were composed of microtubules, and the microtubules in turn being composed of a protein called tubulin protein, they have contracted in such a way as to align the chromosomes right across the middle or equator of the cell. Remember that cells don't have an up and a down, so this line here may not be straight across from left to right. It may be up and down or diagonally, but they will be aligned in a straight line. This is metaphase. In this view of some cells, we have several of them here in metaphase. This cell is in metaphase, and you can see the chromosomes lined up in a straight line here. Here's one where, again, chromosomes don't know up and down, so this line of chromosomes is rather diagonal. Here we have a cell in interphase that the chromosomes are aligned straight across from left to right. These three cells are good examples of cells in metaphase. The next phase of mitosis is anaphase. In anaphase, the chromatids or chromosome strands which arose during interphase separate from each other and begin to migrate toward opposite poles or ends of the cell. The mechanism by which this occurs is not understood. It's been observed, however, that the chromatids must be attached to the spindle fibers in order to effect proper migration. Failure of the chromatids to attach to the spindle fibers results in abnormal chromosome separation and consequent defects in the daughter cells produced. In this view of anaphase, we can see that the pairs of chromosomes have separated so that there is a space in here between the separated chromatids. Now, these spindle fibers are actually pulling on the chromatids and will pull them back towards the centriole. Once that has occurred, then there will be a complete set of DNA at this end of the cell, a complete set of DNA at this end of the cell, and the cell will be ready to pinch into producing two daughter cells. Here's a good example of a cell in anaphase. We can see the row of chromatids here and the row of chromatids here being pulled apart with the space in the middle. Here's another cell in anaphase. The final phase of mitosis is telophase. The conclusion of anaphase and the beginning of telophase are marked by the completion of migration of the chromatids or chromosome strands to the opposite ends of the cells. A cell plate or wall develops across the center of